Okay, in today's lecture, I will be dealing with the tissue engineering aspect. The first, this is like a textbook type of definition. The tissue engineering, the way it is defined, it is the application and principles and methods of engineering and the sciences. So, engineering means it is mostly the biological engineering as well as little bit of the material science. Okay. Biological engineering is already there in the life sciences section. So, life sciences means it is essentially pure biology. Biological and engineering means mostly that engineering disciplines like mechanical engineering, material science, etc., towards a fundamental understanding of the structure function relationship in normal and pathological mammalian tissue and the development of biological substitutes to restore, maintain and improve tissue function. Now, let me explain it little bit more. So, if you remember correctly, in the typical material science, the fundamental concept is to understand the structure property relationship in different metals, ceramics and polymers as well as composites. So, similarly, tissue engineering means you apply the fundamental concepts of engineering that can be either material science and engineering or mechanical engineering or chemical engineering, and life sciences, that means mostly biological approach. And why you are trying to apply the principles of these two disciplines? Your ultimate aim is to understand the structure property relationship in normal and normal and pathological mammalian tissues. Okay. So, basically you want to understand the structure property relationship in these tissues, and if required, with this understanding you would be able to develop some tissue engineering replacement materials, or in other words biological substitutes to restore, maintain or improve tissue function. So, whatever the tissues would ultimately serve certain purpose or certain biological functions, your biological substitute should be able to do those functions. So, that was the ultimate aim of the tissue engineering substitutes. Now, alternative discipline that they have proposed, that tissue engineering can be defined as the application of biological, chemical and engineering principles. So, as I said that engineering discipline means it also contains that material science, chemical engineering, etc. So, essentially here it has been defined as the application of biological, chemical and engineering principles towards the repair, restoration and regeneration of improved tissue using biomaterials, cells and growth factors either alone or in combination. That means, this tissue engineering essentially has been defined as the application of the different engineering disciplines principles towards the repair, restoration and regeneration of the injured tissues. So, basically you can repair the injured tissues as well as you restore the normal functions of those tissues in that implanted area. Now, how you can do? You can use the biomaterials, you can combine use the biomaterials, cells and growth factors. So, these three things if you combine together, then you can actually you would be able to restore and repair the normal tissue functions. So, this is the biological architecture of human tissues and organs. So, if you see, it starts with the fertilized ovum, then in the cell division process and cell growth you are actually replicating the cells. So, you starts with the mother cell, that is fertilized ovum, then it goes to two daughter cells. Now, these two daughter cells in the cellular lineage, then it will be also differentiate or it will be also divided into multiple cells. Now, then cell differentiation means like it involves the changes in gene expression, or it will be transformed from one type of cell to another type of cell. And if you follow this kind of lineage, then you will see there are four types of cells. One is the epithelial cell, another one is connective tissue cells, third one is a nervous cell, fourth one is a muscle cell. And if you combine these four, then you can find that they will form a tissue and finally an organ, and this organ you are talking about, that is that organ system here, urinary system, or organ is a kidney or nephron for example. So, what I am trying to say here, that cells are at the very base level on that cells you form tissues and self organized tissues that will form the organs. So, cells, tissues, organs. So, this is the hierarchical structure of the biological system. So, there are 10 major organ systems in human body. They perform different major physiological functions such as respiration, digestion, etc. And each organ contains many repeating functional units, including a number of tissues like epithelial, connective, nerve tissue, muscle tissue. And for example, circulation system is made up of blood vessels like blood circulation system, which deliver continuous blood flow to each of the functional unit. Now, here is the different cell types in different tissues like epithelia. Epithelia has this absorptive type of tissue like intestine, then secondary cells like planet, beta cell, hepatocyte, etc. Connective tissue is the fibroblast, osteoblast, that is bone tissue, adipose cells, chondrocyte, that is the cartilage tissue, endothelial cells, that is the blood vessels. Nervous tissue, they have different type of cells, again neurons, glial cells, Schwann cells, etc., etc. Blood, you have the RBC, you have these lymphocytes, immune system, you have the monocytes, macrophages, etc. Muscle cells, you have a skeletal, cardiac cells as well as smooth cells, and then you have different cells like sensory cells like hair cells, rod cells, etc., etc. Now, different tissues, they perform different type of functions, because as I have explained to you that in a typically tissue engineering means that essentially you are trying to understand the structure property relationship with different tissues, and thereafter you will apply the concepts of engineering principles to develop biological substitutes, so that it will perform its normal function or restore its normal function of that damaged tissues. Now, therefore, you have to understand that what are the different tissues, and these different tissues what are the functions they perform. One is the epithelial tissue that provides covering, like characterized by the cell shape and then number of layers. You have the connective tissue that provides support. Now, this support is actually kind of different supports, like offer mechanical support like bones, ligaments. Now, if you remember correctly, bones and ligaments, they have very good mechanical properties and originate from the mesoderm, middle layer of the embryo. So, these offer mechanical, so the connective tissues therefore are important because they provide mechanical support. Muscular tissue that provides movement, like responsible for movement like walking, like muscular tissue is also improved for there are 3 types of muscular tissue. One is the skeletal tissue, one is the cardiac tissue, one is the smooth tissue. Then you have called nervous tissue that controls most of the things, like it controls body functions by nervous organs like brain, spinal cord, etc. So, if nervous tissues they are injured, then that will lead to the major deterioration of the body function, as you understand, like if brain cannot work, if the spinal cord cannot work, then that will cause severe problem. Now, these are some clinically relevant tissues by clinically relevant numbers. Now, typically the cell densities per milliliter is type of 1 to 3 billion, that is 10 to the power 9 order cells per milliliter. And if a volume, if you consider that 50 kg of human, that volume is kind of 50,000 milliliter. So, in the 50,000 milliliter, how many cells that will be contained? That is why it has been saying that the human body consists of 50,000 multiplied by 1 to 3 billion cells, and that is roughly around 10 to the power 14 cells. Now, the more the body weight, more is the number of cells. For a child, when the body weight is let us say 3 kg or 4 kg, then you can see that it will be like let us say 1 order of magnitude less, let us say 10 to the power 13 number of cells. Whether if a person whose weight is 100 kg, then you will see that at least 2 times higher than that of the 2 into 10 to the power 14 type number of cells. So, 10 to the power 14 is the typical order of magnitude, that is the number of cells they are present in the body, and 10 to the power 14 number of cells, you can understand that also not a single type, that is also different types of cells, those are present inside the human body. So, it is, as I said earlier, that it is not the magnitude, but it is the order of magnitude which is important, and what the student should remember, that what is the order of magnitude, that is the number of cells that should be present in the human body. Now, a typical organ of 100 to 500 milliliter in size contains typically 100 to 500 billion 10 to the 9 cells, and organs contain functional subunits with linear dimension of typically 100 microns, and the cell number in a cube of 100 micron size would be about 500 to 1000. Therefore, a typical organ will have a few 100 million functional subunits. Now, these functional subunits mostly is related to the polymeric materials or the monomer unit, that is what they are saying, that functional subunits, and typically that 500 to 1000, that is the typical number, that this is the cell number that is there in a cube of 100 micron in size. So, if you consider a typical volume element in the human body which has a size of 100 micron size, so this should have typical cell number 500 to 1000. Now, if you consider that typically a cell as a square like 10 by 10, so 10 by 10 is 100 micron square. Now, if you consider a cube of 100 micrometer size, now typically a cell has a irregular type of structure. So, you cannot consider as a cube. So, it has a mostly it is a spherical like, but it is not spherical. So, it is off spherical, and typically the diameter of the cell is somewhere around 25 to 50 micron. Now, if you consider that little bit higher than the 2 cell size, that is a 100 micron cube size, there you can 100 micron size, then you can consider that there 500 to 1000 number of cells can be squeezed in. So, that is the cell density you can consider at the very finer scale. Now, coming to the bone grafts, now these bone grafts they serve as a structural support and a framework for new bone tissue growth, and allows mechanical force transmission necessary to stimulate new growth. So, bone graft essentially means because of some surgery, some part of the bone is damaged and it needs to be replaced. Now, bone grafting means you are essentially replacing the damaged part of the bone by some new part of the bone. Now, autografts and allografts, these are two biological terms. Autografts mean this is used for small areas of the bone grafting, and here that is bone is still viable, and microsurgical anastomosis of the vascular supply is attempted to increase healing. And allografts mean typically for replacement of larger portion of the bone. So, if it is for the smaller portion, then it is called autograft, and if it is for larger portion, then it is called allograft. Now, this is what is how this tissue engineering concept is applied. For example, now if you consider there is a human being, you take one tissue, you can isolate the parenchymal cells, then you can passage the cells in the culture. So, passaging, those who know about the biology, the passaging means like cells, in order to maintain the cells in a normal healthy conditions, many times you have to drain out the medium and then you have to use a fresh medium. This fresh medium actually provides more nutrients to the living cells, and that are actually used to maintain the living environment in the cells. Now, after the passaging in the cells, then you can select the scaffold or the cell source and seed them. So, essentially then you have to find out. So, if you remember the definition of tissue engineering, whenever you want to develop some biological substitutes in a damaged tissue, then you require a biomaterial, you require cells, and you require growth factor. That is the three things. Now, you have at the stage C, you have the cells, then you require the biomaterial, and this biomaterial is typically, let us say, polymeric scaffold. So, you get this polymeric scaffold, you seed the cells on this polymeric scaffold, and then you can graft them at the sites that where it is required. So, you start with that cells from particular organ, then you go to this biomaterial and you seed them. Then after seeding, you get the tissue engineering substrate, tissue substitute residue, then you implant it. Now, similar things you do it here, as you can see here, that this is a donor. Donor can be one human being, recipient can be another human being, and this recipient is that unrelated to the donor. That means they do not have any blood relation. Now, what you see here, that is a graft site. So, from that let us say you take some skin out of this graft. So, skin graft, then you do some cell culture, and then after the cell culture you can separate the fibroblast and you can separate the epidermis, and then you can put the cell culture and then biomaterial scaffold. Then if you do it after the scaffold, then you can put it in one place. Now, if it is not rejected, that means okay, then you can keep this at that place. But then what has been shown in this slide particularly, that you have the skin graft, okay, and then if you use the skin graft intact, that means without culturing and without seeding on the scaffold, then you can put it on that particular site and there is a chance that it can be rejected. So, what is saying that you can take this graft from one donor, then you have to extract the cells. Like extract the cells means, tissue means what? Tissue is nothing but the self aggregates or the self organized aggregates of the cells. So, you can always extract the cells from the tissue. Then tissue you have to culture them, you have to passage them like this kind of follow this kind of procedure. After you culture them, you can separate fibroblast and you can separate epidermis. Now, these fresh cell lines you can put it on the biomaterial scaffold, then you can put it in the graft material as another recipient. But the point is that if you do not take out the cells from this graft material, then there are chances that it can be rejected by the recipient. So, if it is rejected, then that will cause lot of in vivo reaction to this substrate. So, tissue repair, that has been defined here as the cell based therapies are a distinct class of therapy that rely on cells as the therapeutic agent to affect a pathological condition. What it means? It means that entire therapy, like whatever you have seen, like that constructing the biological substitutes, that is based on living cells. So, essentially you take a tissue from a donor, you extract the different cell lines whatever you require, like depending on fibroblast cells and other type of cells, and then you use these cells as a therapeutic agent to affect a pathological condition. So, that was the overall idea. And the premise of using live cells as a therapeutic agent requires the harnessing of the cell's elegant biochemical machinery to perform functions that cannot be mimicked by exogenous drug products or surgical interventions. So, what has been said here, that the premise of using live cells as a therapeutic agent. So, the cells here it is used as a therapeutic agent in this tissue repair or the tissue engineering, and this requires the harnessing of the cell's elegant biochemical machinery. Like every cell has a particular biochemical machinery. For examples, the mitochondria, that is called known as the energy powerhouse, right, and then you have these ribosomes in the cells which are used for the protein synthesis. Then you have the different other Golgi apparatus and other things. So, they perform different kind of biochemical functions. So, all these cytoskeleton, that actin fibers, they provide the mechanical support to the cells. So, those kind of things, this biochemical machinery needs to perform its own functions and it cannot be mimicked. That means, these live cells biochemical machineries, it cannot be replaced or it cannot be mimicked by exogenous drug products. For example, if a patient requires skin graft, you cannot give the patient some kind of drugs which will replace the necessity of having that tissue engineering constructs. Okay. So, essentially it means that this requirement of having the skin grafts cannot be met by having some drugs or by having some medicines, for example, or surgical interventions, and the use of cells replicative ability for cell production and tissue replacement. That means, what is the next sentence that has been said? Like each cell, they have each living cell, they have a DNA and RNA, right, in the nucleus, and they have the unique ability to replicate. If you remember, the fundamental definition of cell has been said that it is a self-contained unit which is capable of replicating itself, provided it is given the sufficient nutrients and the environment. Nutrients means cells, like a human being, has to be fed some nutrients, and environment means like it has to be kept always at certain pH level, certain CO2 level and certain temperature. If you increase or decrease these things, then healthy state of the cells that cannot be ensured. Similarly, here what has been saying that why that question that comes, that why do we need to use the living cells in the tissue engineering construct? Why can't we replace these tissue engineering constructs with the help of some innovative drugs? The answer is you cannot replace the need for the tissue engineering constructs with some external agents like drugs or some surgical interventions, because one has to remember the cells have the unique biochemical machineries, like different cellular organelles, they perform different things. Okay? And accordingly, those functions cannot be served by some other drugs, that is number one. Number two is that cell has a distinct replicative ability. Replicative ability means cells can replicate himself. Okay. Like one mother cell can give rise to two daughter cells, and each daughter cells can undergo again cell division, mitosis, cytokinesis process, then they can again undergo replication process. That unique replicative capability cannot be met or cannot be provided by any other therapeutic agent. That is why the living cells are used as a good therapeutic agent in this particular case. Okay, tissue repair, they have said that newer cell based therapies that involve significant cellular manipulation in vitro are considered tissue engineered medical products. For example, chondrocytes but cartilage repair. So, many times due to aging and so on, there is cartilage repair is required, and chondrocytes is the cells for the cartilage tissues. Then seeds of dermal fibroblast for the ulcers and burns, and liver and kidney cells grown on extra corporeal support devices. So, these are like three different types of examples that are given. Now, what are the cell sources? Now, the cell sources, the first one is autologous. Autologous means donor back to donor, means you are taking from the donor, you are not using that cells for another recipient, but you are taking the donor cells and you are putting it back for donor cells. For example, suppose a person requires some skin graft at the let us say mouth part. So, what a surgeon can do, they can take out some skin from some tissues from the thigh of the leg, they can culture it and they can use it back in the same person. So, then it is called autologous cells, that means same person to same person. Allogenic means donor to recipient, same species. Same species means human to human. Human belongs to that, if you know biology, human, the species is Homo sapiens. Homo sapiens is the species. Then synergenic means genetically identical donor like homozygous twin. Okay. So, syngenic means genetically identical donor, and xenogenic means that cross species like using animal cells in the human. For example, you take some cells from the mouse or rat and you try to put it in the human being. So, that is like from one animal to another animal, but then it is called xenogenic. Okay. So, these are like biological terms like autologous, allogenic, syngenic and xenogenic. Now, wound healing, that wound, what is the definition? That is created when the anatomic integrity of the tissue is disrupted. So, that is the fundamental definition of wound, and that it said that wound is created at any place in the human body when the anatomic integrity of the tissue is disrupted. Like if you suppose you are falling from a cycle or you are falling from a motorbike, then what will happen? You get wound at some legs or some knee part or something. So, locally at that part of the knee or legs, your anatomic integrity of the local tissues are disrupted, and healing means the process whereby the integrity of the tissue is restored. Like then you put some medicine there, or if it is very severe accident, then your actually entire tissues need to be replaced by some skin graft materials, then it is the process is called the healing process. Now, regardless of the nature, type and size of the wound, the healing process almost universal, and healing is a continuous process with distinct phases that overlap. So, what it says that it does not matter that where you have this wound, either at hand or leg. It also does not matter what is the size of the wound, is it very large wound or is very small wound. The process of the healing is universal. So, this process of the healing will always be followed. It does not matter the place of wound, it does not matter the size of the wound. Okay. So, this is number one point. Number 2 point is that this is almost a continuous process. So, healing let us say has 4 stages, and these 4 stages can overlap. Let us say stage A, stage B, stage C, stage D. Now, stage A can overlap with stage B can overlap with stage C can overlap with stage D, but this A, B, C, D they will follow in a continuous manner. It is not like after A suddenly C will start, it is not like after B suddenly D will start. After A, B will start, but this A and B can overlap. After B, C must start, then B and C can overlap. After C, D will start, but C and D can overlap. So, that is why it is said that healing process is universal irrespective of the nature, type and size of the wound, and it is a continuous process. Now, what is the wound healing? Wound healing is that it consists of 4 basic phases. So, what I was mentioning in the last slide, that is that healing process can contain that A, B, C, D 4 stages. Now, these 4 phases they are hemostasis, inflammation, proliferation and remodeling. Hemostasis is the first phase, it is followed by inflammation, that is the second stage, then it follows by proliferation. Proliferation means you can understand that this is part of the cell fate process. So, cell proliferation, and then remodeling means tissue remodeling. Remodeling means earlier the tissue was in damaged state and now the tissue is coming to a normal functional state. So, that is the process is more or less that can be described as a tissue remodeling. In adults, wound healing process can continue in the remodeling phase for many months. So, basically what is saying that many times depending on the age of the patient, that the tissue remodeling phase can take place for more than 1 month. In embryos, wound healing occurs quicker and without the formation of the fibrotic scar tissues. In embryos, the wound healing process can take place very fast, and in all tissue engineering therapies will invoke the wound healing response. Basically, all tissue engineering therapies, what it means, like therapies means you are again producing the biological substitute to restore the normal healthy function of tissues. So, these all tissue engineering therapies will essentially invoke the wound healing response. So, they will always involve some wound healing response like hemostasis, inflammation, proliferation, remodeling. These 4 stages they must undergo all the tissue engineering constructs. Now, let us go through these stages each one by one. So, the first one is the hemostasis. So, here the platelets means blood platelets they take part. Then fibrin and fibronectin, these are like proteins. So, fibronectin is a protein. Endothelial cells they also take part, and cytokines, and then platelets they are growth factors and cytokines. Then that takes place from seconds to minutes. So, initially you have a wound, within few minutes this hemostasis part takes place, phase takes place. Then comes the inflammation. Inflammation means here the neutrophils, macrophages and then growth factors and proteases they also take part, and that takes part in the case of months to days. So, for few days for example. Then proliferation. Now, proliferation is the place where the fibroblast cells, that is connective tissue cells, and then collagen deposition also take place. So, fibroblast is the very early type of cells which comes in contact during this wound healing process. Epithelial cells and endothelial cells along with growth factors they take part. Then this will be a few weeks. So, from days to weeks. Now, if you see the process, what it says that seconds to minutes means it is quicker, kinetically faster. So, hemostasis process is the very fast process, immediately within the wound, that is the first stage of healing, that is hemostasis. Then inflammation, that little bit slower than the hemostasis process. So, slower in a sense that it takes place till some days. Proliferation, it takes place from days to weeks. Here the fibroblast cells, that is connective tissue cells, epithelial cells and endothelial cells, they take part. And remodeling, you can see the last stage, that is the collagen cross linking. Collagen cross linking means collagen is also, if you remember the structure, the triple helix type of structure collagen, and cross linking means you have the one collagen molecule or collagen fibers and there is another collagen fibers. If some sulfur molecule, sulfur is atom is there, then that will form kind of cross linking between one collagen to another collagen, or there may be some other cross linking agents that is possible use. Then collagen degradation and shear strength and vascularity. Vascularity means there are a lot of network formation through which the blood channels and other things they forms, and that takes place weeks to years. So, kinetically the slowest step is the tissue remodeling, right. Kinetically faster step is the hemostasis process. Is it clear? And then this is the time scale. What is the other thing that you notice that I have mentioned earlier, that hemostasis and inflammation that can overlap. Similarly, inflammation and proliferation that can overlap, and proliferation and remodeling that can overlap. But this is the sequence of events that take place. So, this is number 1, this is the number 2 stage, this is the number 3 stage, and this is the number 4 stage. So, this is the different stages that is present in the wound healing process, and you must remember then all these stages of this wound healing process, they take place in a continuous manner, that is number 1. Number 2 is that all these stages they must take place irrespective of the size or type or place of the wound. So, this is a definition of the wound healing. What it mentions here, that the wound healing is a complex multicellular process with proliferation, migration, activation, differentiation and apoptosis, with proliferation, migration, activation, differentiation and apoptosis of the constituent cells. So, what has been said here, that wound healing is essentially a complex multicellular process. Multicellular means, as we have seen here, blood platelets, that is also one type of cells, fibroblast cells, endothelial cell, epithelial cells, all these different types of cells they participate. Okay. Then it also involves the proliferation, cell proliferation, right. Migration means cells can walk, cells can crawl. This one, cell differentiation process also takes place. Apoptosis means programmed cell death. So, that is also take place. And then wound healing is a process in which epithelial, endothelial, inflammatory cells, platelets and fibroblasts briefly come together outside the normal domain, interact to restore a semblance of their usual discipline, and having done to resume their normal function. So, this is also another unique definition. What has been mentioned here, that it has been mentioned that different type of cells, they come briefly, briefly means for a shorter time period, they interact with each other, and then they will start, all these groups of different types of cells, they will start performing their own functions. For example, connective tissue, that their function is to give support. Okay. Connective tissue cell lines like like fibroblast cells, like epithelial cells, their function is to cover. Cover means to cover the entire wound site from the outside environment. So, similarly muscle cells, that is smooth cells, that also take part. So, all these cells that come together outside the normal domains, they interact to restore a semblance of their usual disciplines. Disciplines means they perform their own duty, and having done to resume their normal function. This brief interaction essentially leads to the formation of a biological tissue there locally, which can perform its normal function. Okay. Hemostasis, that is seconds to minutes. So, that is the first stage. Here hemostasis means to make the blood be still, and disruption of the normal anatomic structure of a tissue damages blood vessels and cells. So, if you see that whenever you have the wound, immediately it starts bleeding. Bleeding means like it starts bleeds, it bleeds like lot of bloods that will come out. Why? Because blood is flowing through a what? Arteries, right. Now, whenever you have a wound, all the arteries they disrupted, they are fractured. So, bloods will automatically come out, right. It will automatically come out and spill. and because anatomical structure of the tissue damages, a number of cascades are initiated, including the complement cascade and the clotting cascade, and these cascades leads to the subsequent migration of the cells into the wound, formation of fibrin clot and constriction of the leakage vessels. Okay. And platelets are trapped in the matrix and release growth factors. Okay. This means that there are different cascades which are formed, and through these cascades that migration of the different type of cells. For example, in the wound place is now exposed to the normal environment. Now, in the wound area you do not have initially fibroblast cells, endothelial cells, epithelial cells. So, those cells has to be migrated to the wound site. So, if it has to be migrated, then it has to be migrated to some channels. So, these channels are formed. So, biologically these channels are formed, and then through that will enable the transport of the different type of cells to the wound site. And the second thing, that whenever the arteries, blood arteries, they are fractured, some constriction takes place. Constriction means at the damaged side, at the fractured side, some constriction, so that this bleeding is stopped and the blood flow, coming out of the blood, that incident is avoided. Now, inflammation, this phase is described by 4 findings, that is redness, tumor is swelling, heat and dolor is pain. So, patient here actually feels the pain because there is inflammation, and redness is caused by the dilation of the blood vessels. Normally you see that at the point of the wound that there is a dilation of the blood vessels in response to the fact that the end products of the some particular acid pathway is blocked, and swelling is caused by the leakage of plasma proteins through leaky vessels, and local heat is caused by increased blood flow and higher metabolic rates. So, increased blood flow actually can cause to local heat. So, locally at the wound area there you will feel little bit hot compared to other parts of the body.